Thank you very much, Professor Law, for your kind introduction. It is actually my great honor to receive this meaningful award from Matthias Day. I'm also so glad to take part in this excellent webinar alongside Professor Jun Chen and present some recent technologies developed in my lab. The title for my today's presentation is Materials Science Innovation in Medicine, Training the Drugs or Therapeutics to be Smarter. And you may be curious and have a question that material science and medicine are actually two totally different fields. How can they integrate with each other? And in our minds, when we are sick, we usually inject drugs and take medicines to help our body fight the diseases. We just need biological scientists, pharmaceutical scientists, and medical scientists to develop new drugs. And why do we need material scientists? In fact, in most cases in the real world, it is not the problem of lacking drugs, but it is a problem that the drugs cannot take action very intelligently and precisely. Two very simple examples to understand this point. We can produce insulin, the most effective protein drug to treat diabetes, but diabetic patients still suffer from many complications because it is hard to control the release of insulin in a real-time manner exactly according to the changes in blood sugar level. Second, many commonly used chemotherapy drugs in the clinic can effectively cure tumors, but cancer is still a major human disease that takes many lives away every year. This is because after administration, the chemotherapy drug molecules would be distributed to both normal tissues and cancer tissues, leading to large side effects and poor therapeutic efficacy. So can material scientists help train the drugs or therapeutics to be smarter and contribute to the medicine field? The answer is yes. I and my team members are actually among such kinds of material science scientists and aim to develop strategies for unmet medical needs in healthcare to improve the lives of patients. The research trends in our lab include fundamental material science innovation, as well as their different applications in translational medicine. As you already know, now it is not enough to just have an effective drug. To enhance the therapeutic efficacy and reduce side effects, the drugs have to be protected and transported to the right location to get an effect at the right time. Therefore, we develop a variety of platform technologies based on biomaterials and utilize them to fulfill on-demand therapeutic functions. Different therapeutic strategies can be designed in different application scenes or in treating different diseases. In a much easier language to understand what we are doing is that we are developing nanoscale or microscale sized spacecrafts or robots to intelligently deliver drugs to the disease sites or directly generate therapeutic functions at the target sites in body, through which we can change the way of disease treatment. Just imagine that if we can effectively control where to deliver therapeutics, when to release therapeutics, and how much and how long therapeutics should be released, we can then simply achieve on-demand therapy with high efficacy and no side effect. This was a dream shown in an old movie, Fantastic Voyage, many years ago. And now we are on the way to fulfilling this fantastic voyage to deliver therapeutics and repair our bodies at the nanoscale or microscale size. Based on the published studies in my team, my talk today can be divided into the following 4 parts: ion delivery, responsive drug delivery, oral drug delivery, and therapeutic materials. The former 3 parts all focus on material science-enabled drug delivery technologies, while the fourth one focuses on biomaterials that can directly generate therapeutic effects without any drug loading. In addition, it is worth mentioning that they can also be easily integrated with each other to achieve enhanced therapy. The first part is our work in ion delivery. Ion-based therapeutics have been demonstrated with promising applications in the field of disease treatment. Their function acts as either an inhibitor of disease protein expression or an upregulator of the therapeutic protein expression. For example, messenger RNA, mRNA, upregulates the expression of therapeutic proteins by direct protein translation on the ribosome in the cytoplasm, while small interfacing RNA, siRNA, downregulates the expression of disease proteins via the formation of iron-induced silencing complex, RISC, and then cleavage of the target mRNA. Due to the poor chemical stability, easy degradation by nucleus, difficulties in intracellular uptake, extremely short half-lives, and other physiological barriers, effective delivery of these iron molecules and making them take action in the right place are among the most important challenges. FDA has approved the use of lipid nanoparticles, LNPs, for safe delivery of iron molecules. For example, the first siRNA LNPs were approved in 2018 for the treatment of a rare genetic disorder, and the recent great success achieved by two highly effective mRNA LNP vaccines during the COVID-19 pandemic further highlights the great potential of nanoscale material-enabled drug delivery technologies. Below is the common structure of the lipid nanoparticles. Basically, the cationic or ionizable lipids are used to absorb iron molecules via electrostatic interactions, and the cholesterol and the PEG layer are used to load and stabilize the RNA lipid complex for in vivo use. And different from this lipid nanoparticle structure, we create the lipid polymer hybrid nanoparticles, LPNPs, for the delivery of mRNA or siRNA in our lab. This new nanoparticle platform uses a polymer instead of lipids to load the RNA lipid complex via hydrophobic interactions, and is finally coated by a layer of lipid PEG to stabilize the system. Such design can majorly have the following advantages. First, improve stability. Due to the solid polymeric core, the LPNP design can provide prolonged protection time for the loaded RNA in the serum compared with the naked RNA and the lipid RNA complex. Second, easy functional design. The polymer part could be easily replaced with functional polymers. For example, responsive polymer that can sense the specific environment in the target sites and enable rapid release of the iron therapeutics only in the right place. I will talk more about this work in a later slide. Third, easy surface modification. Simply by changing the lipid PEG or modifying the terminals of the lipid PEG, different in vivo performance and targeted delivery of iron molecules can be easily achieved. With this universal LPNP delivery platform, we provide the first proof of principle evidence for the restoration of tumor suppressor proteins by the mRNA strategy in vivo. The tumor suppressor gene, TSG, usually acts as either a gatekeeper that can directly suppress the cell growth or a caretaker that can maintain the overall genetic stability of the cells. Loss of TSG function is an important phenomenon in cancer cells, and the approaches for tumor suppressor protein restoration have been demonstrated effective in treating virus cancers in clinic. The current approaches, including small molecule drugs and DNA transfection, although these attempts have shown successes, each has unaddressed limitations. For example, small molecule drugs can be ineffective when TSG has been fully deleted, and DNA transfection has the potential risk of genomic integration and mutagenesis. We can perfectly address both limitations through mRNA strategy because it directly takes action in the cytoplasm and rapidly express proteins without genomic integration. As can be observed in our results, the mRNA-loaded LPNPs have an average size of 130 nanometers, and these LPNPs can effectively deliver mRNA labeled with red color to the cancer cells, while naked mRNA cannot even enter the cells. With an increase in the incubation time, the mRNA carried by the LPNPs effectively escaped from the endosomes, which were labeled with the green color, and entered the cytoplasm to fulfill its function. Moreover, can be observed from our immunofluorescence staining assay, cancer cells lacking KDM6A tumor suppressor proteins could reproduce this kind of tumor suppressor proteins labeled with green color after the treatment with KDM6A mRNA LPNPs, while the empty LP-MPS treatment cannot show the expression of these proteins. As loss of KDM6A tumor suppressor protein majorly occurs in blood cancer patients, we further enable the LP-MPS with mucoadhesive property via amino or sulfhydryl modification on the nanoparticle surface. Through such kind of simple modification with translational potential, the LP-MPS could be administered via intravesical instillation and adhere to the blood wall for sustained delivery of mRNA to the blood tumors. Our results show that the LP-MPS with sulfhydryl surface modification exhibits the highest mucoadhesive property, as the signals that remained on the blood walls were the highest before and after the wash by the urine. With this KDM6A mRNA-loaded mucoadhesive LP-MPS, we successfully expressed KDM6A tumor suppressor proteins in an in vivo preclinical tumor model, where KDM6A gene was fully deleted. As can be observed in the tumor sections, mRNA-loaded LP-MPS treatment effectively upregulated the expression of KDM6A tumor suppressor proteins with brown color, and compared with the control groups, the restoration of KDM6A tumor suppressor via our LP-MPS effectively inhibits the growth of blood tumors, as well as the lymph node metastasis. In the future, we expect that this mucoadhesive mRNA nanotechnology could also be used to specifically upregulate other target proteins within mucosal organs in situ for virus applications. In another work, we further trained the LP-NP delivery platform to be more selective because the concentration of reductive agent glutathione GSH could be about 100 to 1,000-fold higher in tumor cells than in extracellular fluids. We therefore replaced the polymer part with a GSH-responsive polymer to create the new LP-NP mRNA delivery platform. As can be observed from the TEM images and the release profiles, these new nanoparticles showed a fast mRNA release when the reductive agent existed, which particularly benefited by molecules that need to be released into cytoplasma for therapeutic applications. With this intelligent platform, we also pioneered the restoration of p53 tumor suppressor proteins in p53 unexpressed tumors, as p53 off majorly causes the ineffectiveness of many FDA-approved chemotherapy drugs, for example, the mTOR inhibitor everolimus. Our p53 mRNA LP-MPS are like the keys that can turn on the function of p53 and make the FDA-approved drugs work again in treating these p53 unexpressed tumors. Considering loss of p53 function is widely found in many types of cancers, our nanoparticle technology might be extremely useful and widely applied in treating virus cancers. In addition to the above local delivery and systemic delivery strategies for iron molecules, we have also designed different targeted delivery platforms that can home to the specific cells or disease sites for intelligent and precise treatment. For example, for the treatment of atherosclerosis, we designed a target siRNA LP-NP delivery platform, which can home to the atherosclerotic lesional macrophages and specifically downregulate the expression of plaque destabilizing gene, through which the macrophages can be regulated to shrink the necrotic core in atherosclerosis and then blocks the happen of thrombosis. Moreover, the findings herein may shed light on the application of nanoparticle-mediated delivery of other types of RNA therapeutics, such as microRNA and mRNA, to lesional macrophages for the purpose of exploring and validating new targets and pathways in treatment of atherosclerosis. And the second part I would like to share is the responsive drug delivery. Developing stimuli-responsive drug delivery platforms that can sense specific signals and release drugs on demand is an attractive solution to accurately generate therapeutic effects only in the right place. The signals include internal stimuli and external stimuli. Internal stimuli can be understood as special microenvironments in the disease sites, such as pH, redox status, space enzymes, and so on. External stimuli are more easy to be controlled, such as the temperature, NIR light, ultrasound, and the electronic field. These stimuli can also be integrated with each other to generate more accurate treatment. The GSH-responsive LPNP delivery platform for mRNA delivery that I mentioned in the first part of our studies actually utilize the internal stimuli, GSH, within the tumor tissues. And besides the spheroid-shaped nanoparticles, we also prepare sheet-shaped 2D nanomaterials as responsive drug delivery platforms, which can respond to both internal and external stimuli. Due to the ultra-high surface-to-volume ratio, such kinds of 2D nanosheets have extensive surface interaction to enable extremely high drug loading, and the ultra-thin 2D structure can also enable rapid responses to the stimuli for responsive drug release with high sensitivity. These 2D nanomaterials usually have easy degradability and metabolism, which makes them safe for in vivo use. For example, the degradation products of 2D black phosphorus are phosphate ions, which are safe and biocompatible. In addition, the broad absorption spectra from UV visible to NIR of these 2D nanomaterials also lead to excellent photothermal convection efficiency for cancer photothermal therapy and photoacoustic imaging. And as shown in our studies, these biocompatible nanosheets are usually around 100 nanometer in size and several nanometers in height. The drugs with red color can tightly attach to the surface of the nanosheets with black color. And the drug loading capacity can easily reach up to 150 percentages, which is much higher than around 10 percentages for the spheroid-shaped nanoparticles. In addition, they can also rapidly respond to both the NIR laser and the low pH in the tumor microenvironment for triggered drug release, and further enable the deep penetration of the released drugs in tumor sites for enhanced treatment. In another study, we combined the drug-loaded nanoscale materials together with the microstructured hydrogel technology to target the tough chronic wound healing problems. For example, the diabetic ulcer, specifically fibrinogen solution containing pain relief drug-loaded black phosphorus nanosheets were mixed with an equal volume of thrombin solution through simultaneous spray to form the gel covering the wounds. The in situ sprayed gel could act as a biomimic skin to temporarily shed the tissue from the external environment and activate the chronic wound healing, and also acts as a drug reservoir to stop therapeutic black phosphorus and pain relief drugs iodine. With several minutes of the external NIR laser irradiation, the foam gel could generate local heat to activate the microcirculatory blood flow, mediate the release of the loaded drugs for on-demand pain relief, and at the same time kill the bacteria within the injected wounds. And regardless of the different shapes or areas of the wound, the developed technology can be quickly mixed and sprayed at the target sites to form wound healing gels in situ. We also anticipate that this therapeutic strategy may be generalized for treating other disorders as chronic inflammation and infections. The third part is about our works in oral drug delivery. In clinic, oral administration is considered to be the most straightforward route for delivering drugs to intestine and treating intestinal diseases. However, many drug molecules are very sensitive to pH and can be easily denatured and degraded in the acid environment of the stomach, largely reducing their bioavailability in the intestinal tract. Therefore, in the first study, we provided the proof of principle evidence of developing an oral drug delivery platform based on Spirulina-plantensis. We here define it as SP. And SP is one kind of microalga that is edible. Drug delivery platforms based on SP possess the properties of either mass production, and excellent biocompatibility without safety concerns. Moreover, the oral drug delivery platform based on SP have the following advantages. First, the protection of loaded drugs from degradation in the harsh conditions. Second, the intrinsic fluorescent chlorophyll produced by SP allows to perform the non-invasive fluorescent imaging without the need for chemical modification. Third, SP's spiral structure enables it to be not only more easily trapped by the intestinal villi, but also adhered to the intestinal wall, thereby prolonging the retention time of the drugs in the intestine. As could be confirmed from our results, the spiral structured SP have a much longer drug retention time in the intestine compared with the spheroid-shaped particle-based delivery platform. After loading with anti-inflammatory drugs, this platform can also be used for colitis treatment with high efficacy. And in a follow-up studies, we further demonstrated that such kind of SP-based oral drug delivery platform could effectively expand the distribution of drugs from a limited area to a large area of the intestine, which is impossible for commercial capsules. This advantage also benefits from the spiral structure of the SP, which enables them to be easily trapped at the different parts of the intestine. As could be observed from our results, the loaded molecules by SP-loaded delivery platform simultaneously showed full coverage in all the front part, middle part, and the later part of the intestine. After loading AMF, an FDA-approved drug for radioprotection, the AMF-loaded SP could be applied for whole intestinal radioprotection in treating different types of intestinal cancers. Because radiotherapy is one of the most commonly used clinical approaches in treating such kind of cancers, our approach could be significantly helpful to simultaneously reduce the dose and the dosage of the drug administration, as well as the side effects. In the above cases, we hope the drug molecules stay in different parts of the intestine, and in other application scenes, we would much prefer the drug molecules to pass through it and enter the blood circulation to take actions. For example, the most ideal situation for oral delivery of insulin in treating diabetes should have the following 3 steps. This protein drug has to be protected to pass the gastric acid barrier at the first, and once reaching the intestinal parts, the insulin needs to be transported through the intestinal mucosal barrier as the second step. And finally, the on-demand release of insulin should be realized only depends on the blood glucose levels. Therefore, we designed an intelligent oral insulin platform based on a glucose-responsive polymer to load and protect insulin against the harsh GI tract. Immunoglobulin G, FC, was further conjugated on the surface of the nanoparticles to assist in the epithelial transport in the GI tract by specific FC receptors overexpressed throughout the intestine. As can be observed in our results, the FC-modified nanoparticles could overcome the GI tract much more effectively than the non-functional nanoparticles. In addition, the effective release of loaded insulins only happens when the blood sugar level is higher than the normal range. Notably, our oral nanoparticles also extended the therapeutic effect to over 16 hours in type 1 diabetic mice. This platform technology may possibly reduce the frequency of insulin administration to 1 treatment a day. And the last part is about our work in theranostic materials. Different from the nanoscale or microscale materials that utilize the stimuli to trigger the drug release in the responsive drug delivery platforms, the theranostic materials are biomaterials that can directly generate therapeutic effects without any drug loading. For example, biomaterials-based photosomal agents or photodynamic engines can directly generate therapeutic effects through heat or ROS under the external NIR laser irradiation only in the target sites. So one with heat generation is called photothermal therapy, PTT, and another one with ROS generation is called photodynamic therapy, PDT. Such kinds of phototherapy can have the following advantages, such as minimal invasiveness, high selectivity, easy procedure, faster recovery, and so on. Recently, we reported the synthesis of a new kind of 2D nanosheets from vermiculite, VMT, for combined PTT and PDT in one single integrated platform. Through a combination of grinding, catenation, etching, and sonication, the functional core layer, FCL, nanosheets size around 100 nanometer, composed of magnesium and magnesium oxide and aryl trioxide was successfully captured from two sandwiched layers composed of silicon dioxide and aluminum oxide in VMT. It is also worth mentioning that both magnesium oxide and aryl trioxide are FDA-approved materials for clinical use. VMT is also a traditional Chinese medicine, which is included in the Chinese pharmacopoeia. Therefore, the obtained FCL-based nanosheets will be quite safe and biocompatible for in vivo use. These pegylated FCL nanosheets could generate a significant photothermal effect under an HOH nanometer laser irradiation, and at the same time produce massive ROS specifically in tumor sections under another 658 nanometer laser irradiation. As could be observed in the preclinical results, combined PDT and PDT could effectively inhibit the growth of tumors and even eliminate the tumors completely. And the most important thing is that the therapeutic effects only takes place in tumor sites, as shown in the tissue section results. Moreover, such kinds of 2D nanosheets could also be used for imaging-guided combination therapy. For example, we developed a copper iron modified black phosphorus nanosheet-based photothermal agent for PET imaging-guided enhanced photothermal therapy, because PET imaging of these nanosheets could further enable the real-time monitoring of therapeutic responses with a high detection sensitivity. This platform could realize therapy and monitoring at the same time in the treatment of various cancers. Similarly, not only the external stimuli could be utilized, the internal stimuli in the disease sites could also be used for the design of theranostic materials for medical applications. In this study, we prepared arsenic nanodots, ASNDS, from the bulk arsenic materials and demonstrated their selective killing effects in cancer cells, but not in normal cells. And this selective killing effects is caused by higher oxidative stress in cancer cells, which can turn the ASNDS into toxic arsenic trioxide, ATO. And the transformed ATO can effectively decrease SOD activity, leading to massive ROS production, cell cycle arrest, and mitochondrial damage, specifically only in the cancer cells. Considering the arsenic drug ATO is the most powerful clinical drug in treating APL, one kind of leukemia, these ASNDS may have great potential in future clinical use and extend the arsenic drugs application from APL to solid tumors. And finally, I want to thank all my previous and current team members very much for their hard work. Many thanks to the always support from my previous advisors, current collaborators, and co-authors. Thanks very much for the funding support from our department, Stepping Stone Center, Matt Vivo, and many others. And thank you very much for Matthias Day to giving me this opportunity to present some works in my lab. Thank you very much, Professor Low. Yeah.
Great. Well, thank you very much, Dr. Tao. This is a very inspiring talk. I think we are getting some questions from other channel. Let me ask those questions for the audience first. The first question, I believe, is from the WeChat channel, is about how do you actually keep the stability of RNA during the fabrication process?
It actually the mRNA stability can be stabilized by different kinds of chemical modification, such as the cap technology on the five terminal of the mRNA, and I think also the high-tech company TriLink have stabilized such kind of mRNA or sign very stable, and the commercialized mRNA has the advantage of such kind of stability. And during our preparation, the preparation process should be like very immediate after the throwing of the solutions, and it should be a very short preparation method, like within within half an hour, or even we need to keep the preparation time within the several minutes, or even within 1 hour.
Okay. All right. Another question. Maybe I can quickly ask one question about your. I think I believe this. You talk about the RNA, and you talk about responsive RNA-based drugs. One issue with a lot of those external stimulants is about the penetration depth into the body and tissue. I believe you demonstrate the NIR. Is there any other kind of external stimulus this particular LPMP can be functionalized to help? For example, yeah. I know the ultrasonic seems to be a very promising stimuli. Is that possible for your system?
Yes, it is possible. As shown in this slide, there are basically two kinds of stimuli. One stimuli is the internal stimuli. It is majorly based on the disease microenvironment, such as the pH redox and enzymes that are specifically in each kind of diseases. And another stimuli is called external stimuli. That one will be much more easy to be controlled because we can manually control it outside the disease sites, like the temperature, magnetic field, and electronic field, light, and ultrasound. And we developed a variety of materials with a library, and each library can respond respectively according to the stimuli we used, and to realize the function that we need. And the most important, all these materials are biocompatible and can be safely used in vivo.
Okay. There is another question from the channel asking what are the key benefits of using spirulina plantensis for drug delivery in intestinal diseases? Did you get the question? The SP for the
yeah. I think it's all the drug delivery work. Yeah. I think it's a very good question. I think the following points are the advantages of using SP-based oral drug delivery platform. And first, as a natural microalgae, SP is an edible microorganism. It can be eaten. It is just like a sea sage. The Chinese name is hai tai. It is very safe, can be eaten directly. And it also has been massive produced and developed into dietary supplementations due to its richness in multiple nutrients. That is, the drug carrier based on SP possesses the properties of easy quantity production, excellent biocompatible without safety concerns, and potential healthcare benefits. And second, previous works have demonstrated that these microalgae have antioxidant and anti-inflammatory properties, and the regulation of intestinal microbiota, which would be beneficial to the prevention and the treatment of many intestinal diseases. And third, the spiral structure of SP enables it to be not only more easily trapped by the intestinal villi, but also adhere to the intestinal wall, thereby prolonging the retention time of the drugs in the intestine. And also, the intrinsic fluorescent chlorophyll produced by SP allows it to perform the non-invasive imaging without the need for chemical modifications, making SP particularly suitable for both therapy and imaging at the same time. And we can also see it can reach one kind of very unique property that the commotion capsules can't reach, because it can expand the full coverage of the drug in the whole intestine, while the capsules we usually like administrate can only have like part-time appeal of the drug. So when we treat large intestinal inflammation, our platform will be very unique. And even in the intestinal cancers, because the radiation will be a large area, such kind of drug delivery platform can enable a large area of radioprotection during the cancer treatment in the intestinal part. Yeah.
Okay, great. I think we will ask the final question for Dr. Tao, and there's one question for Dr. Chen from the earlier chat. We'll also ask that question. So for Dr. Tao, there is a question about why do you actually choose RNA, not directly use more stable DNA?
Actually, I also have mentioned the advantage of use such kind of technology. Yeah, certainly this is a very good question. And as I mentioned, the DNA transfection using nanoparticles will insert the DNA into the cells, and the DNA may have like integration with the genomic materials. It can be integrated into the cells and make genetic mutations, and such kind of introduction is unreversible, and it will like within the cell reproduction, it can generate to the next generation. For mRNA technology, it just enter the cell cytoplasma, and the expression of the protein is temporarily. It means that when we need it, it show up. When we don't need it, it disappears. So this is a much on-demand technology than the DNA transfection. For example, the cancer vaccine, the COVID-19 vaccine, we just need the mRNA to express the protein temporarily, and the temporary proteins can effective enough to like lead to the immune response inside the body and generate our protection against the virus. And we just need it temporarily appear. We hope it don't last for a long time. Yeah. So for this kind of technology, we can avoid potential risk of genomic integration, and we can need a protein only when we need. Yeah.
Okay. Maybe let's, Jun, you are still there. Maybe there's one question for you earlier. It's a relatively straightforward question. One of the audience were asking you, did you actually make all those textile devices in your own lab, or there's any other way of making them commercially?